Hello, everyone. Welcome to PharmTech's Pharma Talks, where we bring you information on pharmaceutical manufacturing-related topics straight from industry insiders. Today's talk will focus on advanced characterization of sodium alginate raft strength for the treatment of gastroesophageal reflux disease. Presenting today's topic is Ratnakar Palarapu, Analytical Team Leader of Pharma Solutions at DuPont Nutrition and Biosciences, and Dr. Matthias Nahr, Research Scientist of Pharma Solutions at DuPont Nutrition and Biosciences. We encourage you to ask questions during the event, and Ratnakar and Matthias will respond to you at a later date. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. Additionally, you can click on the Resources tab for related materials. We hope you enjoyed today's presentation. Matthias, please get us started.
Many thanks for the kind introduction, Amber. But let us start now with an overview how alginates are produced and some basic introduction to the structure, property, performance of these polymers. Sources for the alginates are brown seaweeds, and the process to make sodium alginates from these seaweeds is quite straightforward. Difficulties arise from the physical separation which are required, such as the need to filter slimy residuals from viscous solution or to precipitate materials which hold large amount of water within their structure. Let us now have a look on the process details. First of all, the source of the brown seaweed has decisive influence on the final chemical composition and therefore on the applied performance of the alginate. As a nutrition and bioscience business of DuPont, we have the unique access to Laminaria hyperborean at the west coast of Norway, which enables us to deliver alginate products with high gel strength. Only 0.3% of the quantity of the seaweed is harvested in a 5-year cycle, which allow us to deliver sustainable products. The harvested seaweeds are first grinded to 5 to 10 millimeter square pieces. In the seaweed, the alginate is mainly present as calcium salt. So the first part of the process is to convert the insoluble calcium derivative into a soluble form, which is described as the extraction process, which is performed with low pH aqueous solutions. The dissolved alginate must now be separated from the seaweed residuals, which is mainly cellulose. The residual is usually slimy and it is removed by filtration process. Now the alginate is in solution and must be recovered in a solid form. Evaporation is not practical because these solutions are too dilute. Therefore, calcium salts are added again, which precipitates the alginate in form of fibers, which are then separated on screens and washed with water. The calcium alginate fibers are afterwards converted to the alginic acid in an aqueous-based ion exchange process. From here, the product is converted to the desired salt form, and for anti-reflux formulations, the sodium derivative is a common one, but all other forms like magnesium are also available. These products are then dried, milled to the desired particle size, and afterwards the products are blended and packed to the customer. These products are natural polysaccharides, which are biodegradable and safe for consumption according to GRAS, which stands for generally recognized as safe. From the chemical perspective, alginates are linear polymers based on 2 monomeric units. Beta-1,4-menurate in the D configuration and alpha-1,4-glucuronate in the L configuration. Both are shortened as N or G monomers. It has been shown via NMR spectroscopy that the polymer is mainly made of blocks of G units, blocks of only M units, and blocks of alternating G and M units. It is important to note that the shape of the M versus G blocks differ to the other. M blocks are formed from equatorial groups at C1 and C4, which leads to a straight polymer with a flat-like ribbon structure. However, the G blocks have axial groups at both C1 and C4, resulting in a buccal chain. This buccal chain are of high importance for the gel formation and the interaction with the calcium ion. In order to achieve the full functionality of the alginate, the product needs to be hydrated in water. These require normally high shear treatment to prevent clumping. When hydrating the polymer, the viscosity is enhanced of the solution. M blocks in the polymer chain are forming random coil structures in solution. The G blocks instead show the buckled chain elements which are able to incorporate calcium ions. This leads to a gel formation based on junction zones formed of 3 to 4 calcium ions with 6 to 8 G monomers. If additional amount of calcium will be added slowly, multi-layer junction zones can be formed over time, leading to further increased gel strength or even precipitation. Let's now have a look on the detailed structure of these junction zones, also described as the egg box model. This model describes a cooperative mechanism of binding between the buckled hyaluronic units and as a two-dimensional analog of a corrugated egg box with interstices in which the calcium ions are based and coordinated. Based on the dimensions in the G blocks between the carboxyl and the hydroxyl groups, calcium ions are incorporated with high efficiency, whereas smaller ions like magnesium are not coordinated that well, and these are forming less strong cells. As described before, N blocks in the alginate are forming random coil structure in solutions. These are forming mostly entanglement and enhance the viscosity. But these M-rich structures are not able to coordinate calcium ions very efficiently and only forming weak gels. Therefore, the source of the brown seaweed is of high importance. The Laminaria hyperborean is a source which offers alginate with high portions of G blocks, forming rigid gels suddenly as the calcium ion concentration is increased. Alginates are used in a variety of different application areas. These take advantage of the strong interaction of alginates with calcium ions. Low amounts of calcium will only increase the viscosity of the solution, which is used for stabilization of juices, dressings, etc. As the concentration of calcium and the viscosity increases, the solution changes from pseudoplastic to thixotropic rheological behavior. Such solutions need time to recover to the original viscosity prior to high deformation processes. Examples for such application are gel structures like creams or others. Further increased amount of calcium lead to strong gels which are used in anti-reflux formulation or for sustained drug release. If the calcium concentration is above a certain limit, the multi-layer junction zones precipitate the polymer from solution, which is used, for example, for water treatment or encapsulation process. Please also note if high amounts of calcium are already present in the water where the alginate should be hydrated, the polymer will not be dissolved in such a media. The calcium source must be added when the alginate is already dissolved in most cases. One of the applications which is of high importance from the pharmaceutical applications are anti-reflux formulations, where the alginate is used as the API. When formulation containing alginates reaches the stomach, the polymer interacts with the gastric juices and forming strong gels which remains in the stomach up to 3 hours. Calcium and or sodium bicarbonate salts in the formulation are forming carbon dioxide in the acid environment of the stomach, which is entrapped in the gel, and the gel buoyant. The gel is therefore floating on top of the gastric juice and provides a mechanical barrier against reflux of the stomach contents into the esophagus. As the intragastric pressure increases, the gel is pushed upwards into the lower esophagus and it's coating and protecting the mucous membrane. With this, I would like to pass it all now to Rataka, who will give you an additional details about our products for this application.
Thank you, Matthias. Coming to protonol LFR 5/60, like DuPont produces different grades of sodium alginate for different applications, and this protonol LFR 5/60 is the sodium alginate grade which is used in the application of anti-reflux suspension formulations, which is like protonol LFR 5/60 is a low molecular weight sodium alginate extracted from the stem of Laminaria hyperborea, which are harvested and produced in the Norway. These grades of sodium alginate has high content of G block with high gel strength. The typical specification limits from different sources of sodium alginates are presented in tabular format, which are used in the anti-reflux formulations are like assay, viscosity, ash content, LOD, etc. And all the different sources has shown similar specifications, except like the color, it varies in the color ranging from white to pale yellowish brown color powder. For the preparation of anti-reflux suspension formulations, the ingredients used are tabulated with their concentration and functions. The key ingredients used in the Raft-formation suspensions are sodium alginate, which acts as a gel-forming agent, sodium bicarbonate, which acts as a carbonate source for buoyancy, calcium carbonate, which acts as a calcium source for alginate gelling, and carbomer, which acts as a suspending or viscosity modifier. Apart from these key ingredients, there are other ingredients which are used as preservatives, pH modifiers, and vehicle. And in the table, we can see like the key ingredients and their role in raft formation are highlighted, and the percentages of use are mentioned in the tabular format. So coming to the raft strength testing procedure. So the raft strength measurement method involves the preparation of raft by using 0.1 normal HCl as per the British Pharmacopoeia. A 150 ml of 0.1 normal HCl is added into 250 ml beaker and kept in water bath to attain temperature of 36.5 degrees centigrade to 37.5 degrees centigrade, and a L-shaped probe which is suspended in the center in the solution with a support. Once the temperature of the solution is reached, maximum dose of suspension formulation is added to the solution. It is kept for 30 minutes in the water bath. After 30 minutes, the beaker along with the probe is removed from the water bath and the probe is attached to the texture analyzer. The raft strength is measured by lifting the probe at a speed of 5 mm per second. The raft strength is recorded in grams, and the minimum raft strength required as per British Pharmacopoeia specification is not less than 7.5 grams. So now moving on to the raft characterization. The suspension formulations which are prepared from different sources of sodium alginate are characterized for the raft strength using texture analyzer. The bar graph on the left side of the chart shows that the raft strength exhibited by the different formulations. The formulation prepared from the protanol LFR 5 by 60 has shown higher raft strength. The images on the right shows the quality of raft formed from the different formulations. The images shows that protanol LFR 5 by 60 has formed uniform raft and which has excellent thickness compared to that of the other rafts formed. We will discuss about the role of key ingredients and their impact on the raft strength. A study has been conducted to understand the key ingredients in the raft formation was carried out by varying the amount of sodium alginate, sodium bicarbonate, calcium carbonate in the suspension formulations. The study revealed that the raft strength increases with increase in the concentration of the sodium alginate as well as with the calcium carbonate by forming a stronger gels. The results are represented in the bar graphs on the left side of the chart. I'm handing over to Matthias for the continuation.
Many thanks. The second method we have used to characterize the performance of the raft was first described by Detmer et al. In 2005. This method has now been further modified by us. The method itself is based on a forward extrusion process and is illustrated on this graph. The geometry is first placed into a hydrochloric acid solution, in which the anti-reflux formulation is poured afterwards. The beaker containing all of these are then stored for 30 minutes at 37 degrees C, and afterwards the hydrochloric acid is decanted through the holes in the bottom plate. The raft gel remains in the geometry and is extruded with a piston connected to the texture analyzer through the openings in the bottom plate. During the measurement, the texture analyzer is recording the force which is required to extrude the gel. Results are given as the work of extrusion, which is recorded as the area of force over the distance in kilogram per millimeter. In this slide, we have first used the commercial available setup, which is given on the left side of the slide. 4 different available ground plates are available for the setup, and the investigated work of extrusion decreases with increasing hole diameter. The commercial available extrusion tube had a diameter of 50 millimeters, and in order to extend the extrusion process, we have constructed 2 new prototypes with 35 or 25 millimeters called MIDI and Mini extrusion cell. As button plates, we have used 2 different setups for both, one honeycomb structure and one sieve button plate. The obtained work of extrusion of the new prototype geometries showed increased reproducibility. The new defined work of extrusion should correlate to the resistance against gastric forces in the human stomach under in vivo conditions. And with this, I would like to give it back to Ratnakar, who will guide you through the rest of our presentation.
Thank you, Matthias. Now I will be talking about the evaluation of mechanical properties of rafts formed by the sodium alginate. The literature suggests that the rafts when encountered with shear forces in the human stomach due to different or various reasons like churning of food contents, gastric pressure waves, penetration and extraction of raft into the lower esophagus. It is very important to evaluate the mechanical properties of the raft like overall strength and recovery during the exposure to shear forces relating to that of that which encountered in the in-vivo stomach. So the evaluation of the raft mechanical properties is done by preparing raft in 0.1 normal HCl which is like by adding 150 ml of 0.1 ml HCl into 250 ml beaker and placing it in a water bath for a temperature to attain of 36.5 degrees centigrade to 37.5 degrees centigrade. Once the temperature of the solution is reached, the suspension formulation is added to the beaker and left for 30 minutes. Later, the raft is isolated from the solution and remains of the solution are decanted. This isolated raft is used for the further testing using the rheometer. So, rheological testing procedure involves the raft is placed on the lower plate of the filter plate of the rheometer where the temperature is maintained at 37 degrees centigrade and upper parallel plate stainless steel geometry used for the testing. The by using this isolated raft, we test for two different experiments like to understand the overall shear strength of the raft as well as the recoverability of the raft. So in case of shear strength of the raft is measured by strain sweep method where the strain percentage is increased from 0.1% to 100% and the response of the raft to increase in the strain is measured in terms of storage and loss modulus. For the second method of recoverability, similar to the earlier procedure, the isolated raft is placed on the peltier plate and recovery of the raft is measured like by using two steps measurements. In the step 1, the strain percentage is increased from 0.1% to 100%, and in the step 2, the strain percentage is reduced from 100% to 0.1%. The response of the raft to the change in the strain, that is low to higher and higher to lower, is measured in terms of storage and loss modulus, which are termed as G prime and G double prime. So the results are represented in this chart. The shear strength of the raft is measured in terms of storage and loss modulus. The overall overlay of the strain sweep curves obtained are shown in the left chart, where the storage modulus of the formulations were higher than that of the loss modulus, and crossover of the modulus was observed around 3% strain. The formulation prepared from the protonol LFR 5/60 has shown higher storage modulus. The higher storage modulus indicates that the increase in the elastic strength of the gel, which can resist to the shear forces correlating to that encountered in the stomach under the in-vivo conditions. On the right side, the results of the storage and loss modulus exhibited by the raft are presented in the bar graph. So moving to the next results part, that is about the recoverability. On the left side of the chart, we can see the overlay of the storage modulus curves for the different formulations which was obtained from the experiment. So the formulation showed the recoverability of the raft to increase and decrease in the strain percentage, correlating to the recoverability of the raft when encountered with shear forces in the stomach under in-vivo conditions. The extent or magnitude of the hysteresis signifies the damage to the internal structure of the raft occurred due to higher forces correlating to that of the in-vivo conditions in the stomach. The results obtained from the recovery experiments are present in the graph format on the right side, where at different strains in the step 1 and step 2 are presented. So in summary, we conclude that protonol LFR 5/60 sodium alginate is produced through a high control procedures. The quality of the sodium alginate used in the anti-reflux suspensions impacts the raft properties. The characterization of the rafts using the texture analyzer and rheometer correlating to the in-vivo conditions shows that protonol LFR 5/60 results in superior anti-reflux suspensions with excellent raft properties. We have a great global application team with in-depth knowledge available to support the customers. Thank you for watching the presentation.